like J.P. Clancy and Dr. Mike Boyle. They are researchers at their care centers. And when they have a great idea, or even somebody who's not at a care center, or a researcher who may be at an institution that is somehow connected to a care center, when they have ideas for research projects and they need to get support by funding, like money to do the study, then they can apply for that support to the foundation. And if it's approved, then they get a clinical research award. So I oversee that process. I'm going to give you a little bit more detail about those kinds of projects. We accept projects on any topic area, not just infection. We see topics that include GI symptoms. We see things that have to do with CF-related diabetes, liver, sexual reproductive health, and mental health. And of course, infection, we do get a lot of that because that is a priority. A lot of the projects that you're going to hear about today come through my award mechanism. I started working on the Infection Research Initiative about a year ago. And today I'm going to give you a little bit of update on where we are. I'm going to also go into detail about those 6 areas of focus and provide some examples of research that's happening in those areas of focus right now. And then I'm going to tell you about the next steps, which is probably my favorite part. So I remember the first time Penelope, my daughter, was diagnosed with Pseudomonas. I was driving home, and I got a call on my phone, and it was the clinic. And anytime you see a phone call from the clinic, you know this is not going to be good news. She was about 17 months, so this was the first time we'd ever experienced this. So the nurse says, she cultured Pseudomonas. We're going to start her on Cipro right away. And then I'm going to get you Toby. You're going to do 28 days of Toby. And then we're going to recheck. If that doesn't work, we'll do another 28 days of Toby. But don't worry. We usually can clear it up with one or 2 rounds of Toby. So she hung up, and immediately I did what she told me not to do. And I started to worry. And I thought, did I not clean the nebs right? Did I not clean my house right? Did I somehow do something that made her get this? I started going over in my mind, where did she get Pseudomonas? Was it that time she dipped her Oreo cookie in the dog water? So we finished the Toby, we went back to clinic, and we did the recheck. And I have the pleasure of being seen at Hopkins, and our doctor at the time was Dr. Rosenstein, who has been doing CF care for over 40 years. He's still there. And I just let it go, and I said, is she going to be sick all the time? Is she going to be in the hospital all the time? Is this the other foot that drops after the diagnosis when everything seems like it's going to be okay? And he goes, we can eradicate this now. We can start the Cipro, and we can do the Toby, and if that doesn't work, we do it again. But we really do see that it clears up most of the time after 2 rounds. And he goes, 20 years ago, we didn't even treat it. They just let it go. And I thought to myself, wow, we have come a long way, but we still have a long way to go. So I want to give you a snapshot of what our funding looks like. And if you want any detail about this at all, if you go to cff.org and go to the research we fund, you can look into any of these areas and see exactly what I'm talking about here. So this is just a snapshot of 2017. And you can see that the big part of the pie, that $39, almost $40 million, goes to treating CF complications. Okay? That's where our infection portfolio lives. So let's dive into that little part of this circle. So of all of the CF-related complication research that we fund, by far the biggest is infections. So in 2017, we were a little over $17 million. And I can attest that this has increased from year to year. Okay? And again, you can get more of this information on cff.org. So like I said, about a year ago, I started to work on the Infection Research Initiative. I was joined by 2 exceptional women at the CF Foundation, Dr. Ann Field, who is back there, and Mrs. Genevieve Mall, who is in our communications department. So we sat down, we got our heads together, and we were like, what are our goals for this initiative? Well, one, we need to communicate what are those areas of research that we need to see to really improve infection in people with CF. We need to communicate that to our investigators. They don't know what to research unless we tell them, right? They come up with their own crazy ideas, but we need to say, focus. We need this. And that was our first goal. But then the second goal was, how can we communicate to you, to the people who are living with these infections, what our research is? It's okay to show that pie chart that says we've done, over $17 million in CF research, but what does that tell you? It doesn't really tell you much. We also knew that the Infection Research Initiative needed to be a comprehensive approach to infection. If we really want to make a difference and improve the outcomes of people who have infections, we needed way more than just new drugs. That's an important part, but that's really just a part of what we need. For instance, are we detecting microorganisms the best we can? Do we have the right technologies? Is there something that we can do that's better than what we're currently doing? We know that certain organisms cause problems, and we know that some are called harmless, but do we know enough about that? Do we understand microorganisms enough to really understand which ones are bad and which ones are good? We also need to understand when we're treating somebody with infections, is this the right treatment? Are we optimizing our treatment approach? How long does someone need to be on IVs? If you have a pulmonary exacerbation, is it 10 days, 14 days, longer? And also, what happens when somebody goes on a modulator? What does infection look like? So obviously, we needed to really come up with a plan that addresses all of these questions. Like I said, it's much more than just finding new drugs. So Anne, Genevieve, and I, we went back and we said we need to meet with the community. So we had focus groups with parents with children with CF and with CF adults. We met with caregivers. We met with the people who are seeing people with CF infections. We met with infectious disease specialists. And then we launched at NACFC. But like I said, sometimes we need to focus those researchers. And that's when we decided, let's put a number to this. It's kind of like a moth to a flame. And this was our flame. We announced that over $100 million over the next 5 years was going to be committed to CF infection. And as soon as we made that announcement, we had so much enthusiasm. Anne and I had calls and emails saying, what are the areas of focus? Tell us more about what you need to see. What are the research questions you want us to ask? We had such great enthusiasm, and we are so excited to see where this goes. So let's go over the 6 focus areas. I'll give you some examples of research that is happening right now. So this is things that are currently underway. And then I'll tell you a little bit more about where we're going next steps. So the first focus area, and I do want to mention, the focus areas are not in order of importance, okay? They work together to really complete the mission of the initiative, which is to improve outcomes. So we didn't want to number them, we're not ranking them, and we will look at them all separately, but together at the same time. So improving detection and diagnosis. Like I mentioned before, do we have the best detection methods? And are there newer technologies that may make it more accurate and easier, and we can get faster results? Another research question we have is, when do we start treatment if we detect an organism? When does somebody actually get diagnosed with an infection just because it shows up on their culture? Some examples of research priorities that we've identified for this focus area specifically is those novel detection methods, way beyond culture. And when I tell you the word culture, what that really means is somebody in the lab taking that swab or taking that mucus from the sputum sample and just growing it on a petri dish and seeing what grows. Now, this is a really effective way to detect microorganisms. But can we get better? Can we do better than that? We also need to really understand what happens when people can no longer produce sputum. You know, this is something that we know is going to happen, especially when the triple combo comes, knock on wood. And we've seen it with folks who are on Kaleido, that they can no longer spontaneously produce sputum to get that sample. So this is also another area of priority for this focus area. So let's talk about what's going on right now. So fungal infections. I get this question a lot. What is the CF Foundation doing in fungal infections? And I'm happy to say we do have research in this area. Right now, at Pennsylvania, we have a multi-center study that's looking at the optimal method to detect fungus. And then what they're also looking for is if there's a specific fungi that may have a negative impact on people with CF. There's so many questions we don't know about fungus, and there is a lot of research that we need to do to address this, but we're getting started. Another project that is currently underway that is much, much larger than that one is one that is looking to standardize diagnosis and treatment for NTM. NTM, I don't think there's a person in this room that hasn't heard of a horror story about NTM. It is an extremely difficult to treat microorganism, and it often results in a rapid decline of lung function. So we have a team of experts at about 8 care centers across the country, and they're really looking to standardize how this is diagnosed and how it's detected. So they're using best practices right now to standardize that culture method. And then what they're doing is looking at a way to standardize or define the response to treatment. And all of this stuff built together, this standardizing of detection and diagnosis, really builds a framework for when we get those new drugs. Because now we have this network of care centers that are experts in this. So when that new drug comes, we are ready to plug in, and we're ready to rock and roll. One of my favorite studies that I have seen come through my program is a new technology called breath condensation testing. I obviously have 2 smaller children that still get that, stabbing swab down their throat. And what this test is, so if you can imagine a cup that's going to go over the mouth and the nose, and then the person breathes for a little bit, and all that breath condensates and is collected. And then they can actually use that to detect Pseudomonas. This is new technology. This is happening right now. It's being tested in Dartmouth up in Vermont, and I can't wait to see where this goes. So developing new treatments is still important. I'm not trying to downplay this, but it's something that we hear most about, right? We hear about the pipeline and we hear about the new drugs. I'm going to tell you a little bit more about our strategy. I'm going to focus more on that rather than exactly what each of these drugs are. So when the CF Foundation looks at new treatments and meeting with industry, we kind of have this strategy that maybe we need to leverage the broader infection community. We don't need to reinvent the wheel here. There are many, many companies looking for antibiotics and antimicrobials. So we need to find those companies that are already doing this and see if they work in CF. And we make it very, very easy for companies to test their compound, their new drug, to see if it will work for people with CF. And the way that we do that is all of our care centers have these biorepositories. So when you have a sputum sample, then it gets collected, we can send those to companies and say, here is a sample from somebody who has CF, who has this specific organism, test your compound on this. And if we see a hit, now we're in business. But we don't need to reinvent the wheel here. We take it and we use our resources to really leverage the work that's being done. Now, all of these companies, they come in through Anfield, not my program. So if you have any questions about what I'm about to show you, she can answer them after, or we can get back to you after the breakout. So JP showed you this slide. And again, it's something that you probably have seen many times. I'm going to go across the top, because I think sometimes that part gets lost. And I think it's really important to understand the steps in this process. So the first part is discovery, and it's just exactly how it sounds. It's just trying to find something that's going to work. And the next step, so if you find something that works and you get this kind of hit, is what we call it, then it goes to the next step, which is the preclinical. I call that the pre-human step, because this work is still done in the lab. And it's really to test that hit, to verify it. Did this hit really happen, or did we just imagine that? And then we may do some safety testing and to see if it's something that would cause more harm than good, or if it actually has some promise. And if we see that the safety is okay and we see that it actually is a hit, then we're going to move it into phase 1. So that's when it first gets into a human. And we do this in a way that really minimizes risk. So it's usually done in healthy volunteers first, very, very small numbers. Occasionally it may be started in a person with CF, but it's very rare, and it's only in circumstances where we feel very confident that there's safety data to support us moving into humans. After that phase, then it goes into phase 2, which is a little bit like, okay, we're feeling pretty good about this. Let's go and get some more folks and try and see if we can get more safety data, and let's see if it's a little bit, if it works. Let's try it in some people who have NTM or whatever infection. And if that all looks great, then it goes into that pivotal phase 3 study. That's when we get as many people that we need to really verify that this is safe and this works. And that's the package. All of the data that we collect that whole entire way goes to the FDA. And that gets evaluated, and then it's, hopefully, in a perfect world, approval. So again, I want to highlight some of the things that JP highlighted during the plenary. We have a lot of things going on with NTM right now. Again, that's an area of priority because it has such devastating effects on people. And we do encourage development of multi-organisms, a development of antibiotics that will address multiple organisms. All of this information, if you want to hear about what the status is of all of these, go to the drug development pipeline on cff.org. You can literally click on the drug, and it'll give you results if it's available, or it'll tell you where it is, enrollment, what phase. It's a great resource. If you think that there's something on here that might help your loved one or help you, you can enroll in a clinical trial. You can go to the clinical trial finder on cff.org and look for anti-infectives right now that are currently enrolling. So the next focus area is optimizing current treatments. So when Penelope was diagnosed with Pseudomonas, we got a plan. We had a plan. And that didn't come, overnight. That was from research, from our investigators, from our physician scientists who are in the clinic and have to come up with this evidence-based support to help with this treatment plan. And we need more of that. We need to be able to give our CF clinicians evidence-based information on the best way to treat infection. We know that antibiotics can cause harm, so we try to use them as less as possible. We want to reduce that risk. But we also want to make sure that we maximize their effectiveness. So some examples of research we have going on right now is with MRSA. So we are working on, well, the researcher is, I really just kind of see if she needs any help with anything. She is looking for a way to standardize treatment and possibly eradicate early infections of MRSA. Now, this is research that's been going on for years and years and years. And it's still, going to be a little while before we get good information and data back to our clinicians. If you think about clinical research, the investigator side, where it's not really an industry or a corporation behind it, it kind of runs along that same pathway of discovery and phase 1 and phase 2. But it's worded a little different. You might start with a pilot study or a pilot and feasibility study, which just looks at a small number of patients and see, does this work? Are we asking the right questions? And then if you find something that seems to be promising, then you might go into a larger clinical study. So we're in that stages right now where she's done her pilot data. She's got really good evidence, but she's really dialing in what's that optimal protocol? What's that optimal plan? Another research program that we're supporting that I hope you guys have heard of is called STOP2. So STOP stands for Standardizing Treatment of Pulmonary Exacerbations. This is a huge study. So if you're thinking on that drug development kind of line, this would be more towards the end of, we are very close to having some great data to pass out to our physicians. And the STOP2 study is looking at optimizing treatment for pulmonary exacerbations. So when somebody gets admitted to the hospital, and we kind of have an idea of what that's going to look like. But the number of days that you're on IVs kind of varies depending on which hospital you're at. And what this study is looking at is to kind of design a treatment plan that is broad enough that can be applied to many, many people, but still has components that are individualized. So you can say, if this person is responding like this, we're going to do a few more days of IV antibiotics. If this person's responding like this, then we feel like they might need this much. So that study is huge, like I said, and is still enrolling and still underway, but we're hoping to see results soon. And the really exciting news is this STOP program is actually trickling down now. So we're going to be starting a PEDS version of this. But instead of IVs, it's going to be orals. And if you have children that have, young children that have CF, you understand that sometimes treating pulmonary exacerbations in the very young is a little wild, wild west. You don't know, are you supposed to start antibiotics now or not? And you just do. So that really is going to help us answer that question. So the next focus area is understanding CF microorganisms. CF microorganisms and infections, so easy. It should really be solved by now, right? CF infections are very complicated. We have a lot to think about. We have a lot of questions to ask in this area of focus. We really want our investigators, and this is more focused on the basic research side, so not so much in the clinic, but more like in the laboratory, where we really need to look at how these microorganisms interact with each other, how are they acquired, and how our body and our immune system may respond to them. These projects tend to involve a very new technology that's called whole genome sequencing or next-gen sequencing. And what that is basically a way to detect everything that's there. So where the culture, you take a swab, you take some mucus, and you grow it on a culture plate, and you're looking for a very specific bug, right? You have different culture medias that grow different things, so you have to kind of match what you're trying to find with what you're using. But this new technology really just gets it all. Gets it all, whether it's good or bad. And you get a lot of data. So this kind of research right now, these technologies are really for research purposes only. Will they eventually make it to the clinic? Absolutely. But we need to really understand what this data means before we can use it. It's also very expensive. Good luck getting that covered. So, some examples of research that we have in this area is understanding the microbiota of pediatric CF patients, and what role does it play? Microbiota, microbiome, these are really fancy words for every single type of bacteria or you're seeing growing, whether it's good or bad. So we know that there's microbiota that are harmless, and well, what we consider harmless, especially in young kids. But we don't really understand what that harmless bacteria means downstream. For instance, that harmless bacteria may trigger some sort of inflammation. And we need to understand that. So even though this bacteria may not be pathogenic, meaning it might not cause an infection, it may cause issues. So research in that area will be really helpful in understanding when we see this bacteria, especially if we get to these new technologies that capture everything, which are the ones that are really causing issues, directly or indirectly. Another study in this area is looking at the connection of the different types of organisms and trying to connect that to clinical outcomes. So if you can imagine, and I hope this analogy works. So if you imagine a fingerprint, your lung microbiota is a fingerprint. So we do these next-gen sequencing and we get this fingerprint. And we're trying to connect that to clinical outcomes. So people with this type of fingerprint might have less CF lung disease. So we can say that that kind of combination of microorganisms in the lungs is not so bad. We can kind of tailor our treatments around that. So this study is really looking to improve our understanding of how these bacteria interact, whether we call them harmless or not, and really identify any connections they may have with clinical outcomes. So the next focus area is evaluating long-term antimicrobial use. People with CF are on a lot of antibiotics, and we know that this can cause issues. We know that you can have kidney disease or kidney injury from antibiotic use. We know that you can have hearing loss from certain antibiotics. So one example of research we have going on right now is looking at the impacts of prescribing antibiotics for viral infections in young children. That's best practice right now, right? We feel like that's really the best way to treat young children. But we need to really study this and look at the effects it may have. Another really interesting study is to find ways that we can predict individuals who may have more kidney injury because of antimicrobial use. So it would be really helpful for our doctors to know that person might have more severe kidney injury if they take this antibiotic, because that will help tailor their treatment. They might decide to change an antibiotic, they might shorten the length that they're on the antibiotic, or it might change their dose. So being able to identify those individuals would really reduce their risks. And we know that hearing loss is a major concern in the CF community. We currently have research funding on this in both the industry side and the clinical research side. So finally, the last area of focus is understanding how infections are going to look when we have treatments that address the underlying cause of CF. We know from studying folks that have started Kaleido that infection can look very different. And we know that it really depends on at what time, when they start those modulators. If it's very young, infection looks very different than somebody who is older and has more advanced lung disease. We really need to understand this because this is our future, and we need to get ahead of the game. And it's more than just modulators. As you heard in the plenary, mRNA therapy is already in phase 1 studies. And what does it look like for infections when somebody has mRNA therapies? And so on. I mean, gene editing, we'll get there soon. So this kind of research needs to start happening now. So when we're there, we're ready to go, and we can tackle these problems. So one of the largest studies that the CF Foundation has ever funded for investigator-initiated research is the PROMISE study. And the PROMISE study is going to look at people who are about to start the triple combo and then follow them for an amount of time after they start the triple combo. The PROMISE study is looking at so much more than just infection. It's going to be looking at CF liver disease, CF-related diabetes, GI symptoms. It's also going to be looking at how somebody can clear their mucus. But infections is a really big part of this. So some of the things that the PROMISE study is going to look at is changes of baseline for those who do have Pseudomonas and have staph. Can we see a reduction in the amount of bacteria that they have? And we're going to do this in a way that really we can quantify. So we're going to use those more advanced technologies for this. And we're also going to look at the microbiome. What are the changes in just everything that's growing? So it doesn't really matter if it's a modulator. This is the research that needs to happen now. So we had our 6 areas of focus. We got the flame to draw the moths to, so now they know what they want to research. But we need to find a method to make sure that we continue to understand what the priorities are and to make sure that we're on the right track. And that's why we put together the steering committee. So the steering committee's job, and I can give you a little bit more information about this, but the steering committee's job is really to look at our current clinical research portfolios, our industry research portfolios, and our basic science research portfolios. And tell us, are there any key questions that we're missing? Where are the gaps? Where do we need to focus? Where do we need to tell our investigators, okay, look now at this? And as we get research and as we continue to fund, they'll reevaluate, and then they'll advise us if we need to change our priorities. I'm so proud that the steering committee is 1/3 people with CF and parents. This is huge. So our 7 clinical researchers and clinicians come from a broad background. Some are infectious disease specialists. Some are clinical researchers, so we're seeing your patients that are in your clinics. And some are just bench scientists, which, that's really hard to get them out of the lab, but they're willing. And then we have 4 CF community members who are joining us. Ella Balassa is an adult with CF. Mary Lee Phillips is another adult with CF. Jennifer Spalding is a parent, and Kirsten Kulik is also a parent. So we knew we wanted to have a huge community role for the steering committee. We make sure that we have patient engagement in everything we do. And we just told Community Voice we want to find some folks to sit on the steering committee. And it's a 2 to 3 year term, so we will be putting out a call for application again in the future. But we have never had such an overwhelming response for community members to join a steering committee. In the very short amount of time that application was open, we had over 50 applicants apply. It made Anne, Genevieve, and I's job very, very difficult. But we had some ideas of what we were looking for. We needed our CF community members to be really engaged and be able to represent a broad spectrum of people with CF or children with CF. And we also wanted to make sure that they could tap into the community, really get some answers for us, and feel comfortable bringing that back and responding and answering questions with the committee. So we narrowed it down. We did interviews, and we selected these 4. Our first meeting is April 11th, so I'm going to go home and get back to work. I have things to do for that. But we're very excited to kick this off. I'm very excited to hear what kind of recommendations they're going to provide for us and see what happens. So again, I just want to go over those 6 areas of focus: improving detection and diagnosis, developing new therapies, optimizing current therapies, understanding CF microorganisms, evaluating long-term antimicrobial use, and the future of CF infections. We truly are coming from all angles to improve outcomes for people who have CF infections. We might not get to a place right away when the nurse calls and says, "Don't worry," but we will get there. So with that, I'll take questions. So I have a mic, Ann has a mic.
Yeah, I'm going to try and play Vanna White.
And if you're listening in online, you can also submit your questions, and they will get beamed to me somehow.
Thank you. I think I speak on behalf of all of us in thanking you all for planning this committee in such a comprehensive way, in such a detailed way. The outcome is bound to be better. I think even at this table, we've discussed our children we have older children with cystic fibrosis, and now we're running out of arrows in our quiver. So the last time my daughter had an infection, her doctor said, well, we don't want to use the big guns yet because we don't want her to become resistant to that as she has to the current therapies. So I was so grateful that you had this. This question dates back really 20 years when our daughter had a PICC line after a surgery, complications from a bowel obstruction, and she was in the hospital for over 2 weeks. She was in for like 20 days for that period, but after about 2 weeks, she began running a high fever, and it just so happened it was one of those times when the staff change took place. It was around July 4th, and all the people who had been working for, almost a year went on to their next position, and we had the newbies. And so it was nighttime, and Margaret's temperature went up, and the nurse came in and said, oh, I think this thermometer is broken. And I said, no, it's not broken. Her temperature's going up. And the resident, I think, said, she might have a fungal infection in her PICC line. I'm going to check on that. And sure enough, she did, and they gave her diflucan, which is an antifungal drug, and she got better. But it was a long night. It was one of the scariest nights I've ever spent with Margaret. But since that time, I've kind of just stalked some, I'm sorry if I've done this to y'all, but stalked some CF Facebook pages, just kind of listening to what people say. And a couple of parents have said, gosh, my child is in the hospital, has a PICC line, has had it for a few weeks, and her temperature's going up. And part of me wants to go, check it out, see if it's a fungal infection. But I don't, I'm trying very hard not to be that person. So anyway, I wonder if now one of the protocols from, one of these studies is checking on that type of thing, because a lot of our kids or family members will probably end up having a PICC line at some point, and maybe for 2 weeks. And it just seems like this might be just one little area that could save a lot of, pain and suffering and, who knows, a life. Is this a part of the protocol now to investigate that?
Yeah, thank you for that question. It absolutely is part of this initiative. And in fact, we do, I recently did just see a project come through and was funded to look at PICC line and risk of infection. And that's enrolling over 1,000 patients. And if you want more information about that, we can connect later. But that is absolutely something we're looking at.
Hi. I have 2 CF kids, 24 and 21. It seems to me like one of the weak links in infection control is, aside from the medical community, it's how the hospital itself is cleaned. And I know those the folks that clean the room are always the first people that I befriend and make sure are very attentive in our room. In my professional life, I work for the United States Environmental Protection Agency, and we have an entire pesticide program, which is our working with pesticide companies that kill not only bugs and things, but also bacteria. And I always wondered why the foundation didn't reach out more to government to talk. I mean, we have whole labs that study resistance and Pseudomonas in particular, and I've never seemed to get anywhere with getting interest from the foundation and talking to them. So I would love to reach out to you and see if I can connect our folks with your folks.
Yeah, Anne, do you have anything to add to that?
I think that's a really great perspective, and I think one of the things that I'm trying to do is really expand into other people that are doing research in this area. So I would actually love to connect with you and hear about what are those resources, how can we tap into those and learn, because infection, the good thing about infection is it's not our problem to solve alone. There are a lot of other people working in this space. And so I would absolutely love to hear more about that afterwards.
Like you, I clearly remember the day that I got a call from my son's physician. He's now 43, but that was 30 years ago. So they didn't treat Pseudomonas the way they do now. Is research going on to address the continuous culture of Pseudomonas and the biofilm, I think, that occurs in the lining of the lungs?
Yeah, so I mean, we do want to understand, how can we eradicate, and that is an area of focus. And we do want to make sure that we engage researchers who are looking at ways that we can decrease that bacteria load and biofilm. And Anne can talk more about the industry side of maybe some therapies that address that biofilm.
Sure. So biofilm itself is actually a really hot topic right now. There are a lot of companies with different technologies that are looking at breaking up biofilm. So it's something I'm hearing a lot about right now. And so I think as those technologies advance, we'll have more arrows in our quiver around the biofilms. I think, to your point about continuous culture and identifying if there are biofilms there. I think certainly there's the culture guidelines already in place, but that I think, continuing to evolve and continuing to look at how we're doing things, we can always improve, I think is certainly a part of what we're trying to do.
You talked about the modulators and how infection will look, I guess, downstream. Is there, I guess I'll discuss Kalydeco because that's historically the drug that, the CF patients have been on the longest. Is there any data at all about a patient who was infected with Pseudomonas or MRSA and after, 5 years of Kalydeco now it's eradicated, self-eradicated?
Yeah. So unfortunately, we did the study similar to like the Promise. We did that with Kalydeco, and we tend to compare the triple more with Kalydeco because it's on that level of effectiveness is what we're thinking. Knock on wood. And unfortunately, there were some studies done in Ireland that was done in older patients, and when they started Kalydeco with established Pseudomonas, they did see initially a decrease in that bacterial load. So the results of them taking a Kalydeco did see less of that Pseudomonas in their cultures. Unfortunately, after about a year, that bacterial load came back. So there's still a lot of things we don't understand, and there's still things that we need to study. Now, that's just an observational study. So that was just following patients that were on Kalydeco in Ireland. So that's a very small number of people. We do see that people, that younger population that started Kalydeco did have a better chance of eradicating Pseudomonas. So I think it is going to go back to that. When are you able to start that modulator, at what age, and what is your established disease? And it's really going to be an individual story with that, I think.
Have we had any questions come in the back? Okay.
Okay. Wow. So I must have done a really good job explaining everything. All right. Thank you.
Can you just speak for a quick second about the NTM infections, and when do you choose to treat them or not treat them?
Yeah. So NTM is a major priority for the CF Foundation. And in 2017, we established the NTM Consortium. So that's those 8 care centers are the leading experts in NTM research. So together they're developing, and it's actually 2 studies that are going along simultaneously. The first one is called PREDICT, and that's really to establish that protocol for detection and diagnosis. So they're using best practices right now. So they're using the technology and the best way that we know to detect NTM now. And they're discovering that they can find a way to standardize diagnosis. So we know that not everybody who cultures NTM has NTM lung disease. You actually have to meet certain criteria to get that diagnosis. So that PREDICT study is really standardizing that. So that can be applied to everywhere, to all care centers. That's the first half of that. The second half of it is the patient study, and that's really looking to find the optimal treatment. And what they're doing is they have it's called a treatment algorithm. So they start, somebody who is diagnosed with NTM lung disease. So we've gotten to the detection, the diagnosis. Now let's think about how we're going to treat that. And they start them on one path. And if they find that they're responsive, then great, they stay on that path. But if that path doesn't work, then they'll say, okay, let's try this drug. And if that doesn't work, okay, let's try this combo. And it's almost like a decision tree that really is super easy for all pulmonologists at any care center to follow. But it's these leading experts in NTM that are developing it. So this is ongoing right now. And if you have a child that has cultured NTM or have you cultured NTM, you can enroll in this kind of study. And there's a lot of things going on around this consortium as well, including new drug development. We have clinical studies going on some new drugs that may treat NTM lung disease. And then there's also some other research like programs that are using this network to really help understand better this NTM infection.
You said that, oh, I'm sorry, the cup that is being, it was being used to collect bacteria in your child.
Yeah.
Has that reached a point where has been approved or not?
No,
Okay.
so this is very early. In fact, the researcher is doing so well that she actually got NIH funding to boost her research as well. So we gave her support originally, and now the NIH has really thought that this is research worth funding as well. So this is really in the stage where it's validating that this can be done. And once we get those numbers, once we understand that, yes, what we're seeing, these results are true, then she'll take it to the next level.
Back on the infection control thing, one of the things that always frustrates me in clinic and in the hospital is that there seems to be such a wide variability in what's recommended for how to protect yourself in that environment. Like some places, you see kids with the VOG mask, and then you read that the VOG mask is actually bad, and then you have people with paper masks walking around. If somebody puts Purell in front of me, I tell them that the efficacy tests that we do at EPA say it fails 50% of the time. So if you're my doctor, you're going to be using soap and water. And yet I've had doctors say, well, that's not, how are we going to get to educating our own CF care centers as well as hospitals in general?
That's a great question.
About how to do this properly, because right now, I know in my own daughter's chart, it says that I am germ phobic. And I wear that proudly.
But you're in good company. It's a great question. And guidelines are developed by the experts in the area getting together, and they review the literature. They review those articles that your clinical researchers are writing and they publish. And that's really what's needed in order to get those guidelines. So that goes back to that area of focus where it's understanding microorganisms and how they're acquired. We need so much more research in that area because that will answer those questions. And once we have that literature, then we can create those guidelines. I wish it was a faster process because I'm right there with you. I'm like a hawk. If I see somebody come into my child's hospital room and they're not in that droplet gear, then they're going to hear from Mama Bear. And and we know that, but those guidelines, they take time. And then they need that research to back it up.
It shouldn't be our job to be that person. It should be the clinic's responsibility to understand. I'm sorry. It shouldn't be our job to enforce that.
Yeah.
Some of us are going to do that because I'm from New York, and you can all tell I'm very assertive on things. But I don't really feel like that should be my role. That should be the clinic should understand that they're professionals. Okay.
Yeah.
And maybe we can come up with interim standards that are better than what we have right now, which is nothing.
Yeah. Oh, we have a question on. Anne, actually, I think this is a question for you. So, do you want me to read it to you, or? So, is there any treatments other than vancomycin that is being researched to help treat MRSA and keep it away permanently? Yes, I think this is a trick question.
That's a trick question, yeah. So yes, there are other things that are being looked at to help treat MRSA. Where we have I see things coming through. Nothing on our funded portfolio other than inhaled vancomycin right now. But certainly there are other things coming through the pipeline to help treat MRSA. I think keep it away permanently is the part that's the trick question. I don't know that any infection treatment can keep an infection away permanently. I think there's always that chance that you'll reacquire it, and that's why I think we need to have that continuing pipeline to continue to get those new arrows in our quiver, new things that we can try and use. What I am excited about is that a lot of the approaches we're seeing now are novel approaches. They're not classic antibiotic approaches, and so that's exciting because it means there are different mechanisms that the bugs may not have developed resistance to yet. And so I think it's a really exciting time, and so I think we'll see lots of new things coming through in the near future. And Dara, we have one more question that's come through online. Are there studies on the effect of diet on symptoms?
None that really come to my mind right now, off the top of my head. But that certainly doesn't mean that we won't ever see them. We do see a lot of nutrition projects come through. Now, typically these have to do with improving nutrition for people with CF. And I guess one could argue that if you improve nutrition, then you can also improve a lot of other things, a lot of other clinical outcomes. But nothing specifically, unfortunately. Okay. All right. Well, if there's no other questions, then I really want to thank everybody. This has been such a great VLC. I'm partial to this conference. I'm a little biased. But thank you for joining us today, and I hope you enjoy the rest of your afternoon.